Hi, I'm Kishore from Explore CSAI, and my project is on the topic AI in protein sequencing, structural prediction, and binding site finding. So, as we all know, protein has a very complex structure in nature, and it is formed of amino acids. So, previously we used to find the structure of a protein with help of experiments. Like we had experimental methods such as X-ray crystallography, which tend to be more costly and labor effective or labor intense. So, recent developments in the areas of AI and deep learning technology can help us overcome that issue and find out or come up with a more efficient method to find the structure of protein. So our aim is to find structure of protein by predicting the folding of protein with the help of deep learning tools such as AlphaFold, which uses attention-based convolution neural networks, and to also find the binding sites of those protein structures with the help of existing bioinformatics tools from Python. So we'll see in detail in the upcoming slides. So this is the brief introduction about our project of like what we are going to do and what this is about. Our project is divided into two parts. The first part is predicting the structure of the protein, and second part is to find the binding sites in that structure. For the first part, we are using a deep learning tool called AlphaFold that uses attention-based neural networks and transformers to predict the structure. So after finding the structure, we are using bioinformatic tools such as voxels. Voxels is like a 3D pixel, which helps us to find the gaps in those structures, those gaps where ligands and drugs bind to. Those gaps are also called as pockets. So our aim is to find the most probable pocket, or the pocket with high chances of drug binding. So overall, this helps in drug discoveries and helps us to find target binding site of a drug. So protein is very important as it governs everything from cellular function to disease mechanisms. And understanding structure is crucial for future drug discoveries and uh, improvement in biotechnology. So existing methods, as I said, tend to be more costly and very time consuming. So before 2020, when we are still using experimental methods to find the structure of protein, uh, we were only able to find structure of 100,000 proteins. And by 2022, when AlphaFold2 came into action, which uses more advanced technologies such as Evoformer and everything, we were able to predict structures of over 200 million proteins, which is a great improvement as compared to previous stages. So we are trying to implement that technology and to find drug binding sites to those structures predicted by our existing deep learning tool. So our primary objective is to develop a hybrid analysis pipeline or a hybrid workflow between two different technologies, which is one is deep learning or AlphaFold, which is used to predict the structure of the protein, and another one is manual method of finding the binding sites. We are using spatial voxels here, and to combine those both and to create a seamless workflow between those two. Like we are inputting a protein sequence and we get the complete analysis of what the structure of the protein will be like and what are the possible binding sites in that protein, etc. So we are also trying to implement what are the possible drugs that can be binded into that protein, but we are not so sure about that as it requires much more data about drugs and uh, those binding sites of those specific drugs. So we are trying to study more about protein because, as I said, they play an important part in any living organism. They they act as enzymes, provide structural support, they signal to our systems, and they also sometimes act as immune defense or antibodies. So they are very crucial, and finding about them or studying about them more will only help us. And we already found methods to find the sequence of the protein, such as Edman degradation, which is a traditional method that we were using for ages, and we were also using spectrometry, and we can also find the sequence of protein by DNA translation. So, why do we need to predict the structure of the protein? Why don't we just uh, find out experimentally? As I said, experimental methods are tend to be more costly, and many proteins are the structures of many or the sequences of proteins are already found, but the structures have not been predicted yet. So that's where deep learning methods come to play and shows themselves as much useful than experimental methods. So we started computation methods before 2018, which started from homology modeling, which we use the existing protein structures to train a model and find a new model, which is not much effective, and the error rates were so high. And there comes ab initio modelings, which is like instead of using already existing structures, we use the physics principles of folding in a protein, and we used to predict the structure that way, which uh, interestingly made a much more impact than homology modeling. And after that, AlphaFold2, which uses much more upgraded things such as Evo former and structural modules to predict more than 200 million structures. So that uses AlphaFold. And before AlphaFold, I mean, this is used with this deep learning method is used by AlphaFold2. And before AlphaFold2, there were like Rosetta Fold and AlphaFold1, which used ab initio modelings that use the physics-based theories and physics principles to find the protein structure. So, what are binding sites? Binding sites are the regions in the protein where protein interact with ligand, DNA or RNA molecules or drugs or other protein as well. So there were other experimental methods to find the binding sites such as X-ray crystallography and NMR spectroscopy or nuclear magnetic resonance spectroscopy. As I said, these are experimental methods, and we can always come up with new computational methods which will be effective than these methods. So some of the computational methods are geometry based, which is we are doing right now using voxels, and there are other methods like CAST-P, Pocket Finder, and we also have machine learning based models such as DeepSight or ScanNet, or energy based, which is Autodoc and SiteFound. So to predict the structure of the protein, we input the protein sequence as a FASTA file, which we can get from databases like UniProt or any other publicly available databases. So we feed that protein sequence into the uh, AlphaFold model, and the AlphaFold uses multiple sequence alignments, evolutionary data, like which is the past data of the protein's evolution, and attention-based neural networks to give the output structure as a PDB file, which can be later used in predicting or finding the binding sites of the protein. So to find the binding site of the protein structure, we are using grid-based spatial modeling. So here we create a 3D voxel around the protein structure and map the atoms into that. And after mapping, we are inverting that structure. And when we invert, we will find the cavities or depressions in the surfaces. So those are the pockets in that structure. We also have a certain threshold to avoid biologically irrelevant cavities or biologically the cavities that doesn't contribute much to the uh, binding or anything. So after finding such depressions or pockets, we analyze the nearby residues and uh, score them based on their spatial proximity. So residues means amino acids in protein structure. So after scoring them, we can find which ligand or which pocket is the highest probable for a ligand to bind. So as for the updates, we are not yet done with the first part of the project, which is implementing the AlphaFold and finding the structure. And we are almost done with the second part of the project, which is binding site finding. So we need protein structure to find the binding site, which is uh, retrieved from the first part of the project, or the AlphaFold. Since we are not able to complete that first, we are using online available PDB files, and right now we are able to retrieve all the pockets in that specific protein structure, but we were not able to score them based on the probabilities, which we will be doing as the next step of our project. And we are trying to implement or trying new ways to implement the AlphaFold 3, which is the latest model of the AlphaFold, and find the model. So we're still working on that, and we are expected to complete that work by next month or end of the next month. And after that, we still have to check and check for errors and everything, and we might have to fix every errors and problems in the code. And at last, we are trying to develop this as a web application so that we can get input from protein sequence input from the user and analyze the protein and give them every data of what we know till now. So to summarize, our project has two parts: prediction of the protein and finding the binding site. To predict the protein, we are using AlphaFold, which is a deep learning model. To be precise, we are using AlphaFold 2. And for the binding sites, we are using geometrical based bioinformatic tools available in Python library. In our case, we used 3D voxels. We used 3D voxels to find the cavities in the structure in which the ligands or drug bind to. And that cavity is also called pockets. So our aim is to seamlessly connect or create a workflow between the AI-driven architecture and uh, the Python libraries such that we can predict the structure and find the binding sites of a protein sequence. So basically, we input a protein sequence as a FASTA file, and we our system gives you the details about the structure of the protein and most probable binding site of that protein sequence. So that's it for our project, and I have also attached a video, a working demo of the second part, or as the binding site of the protein. I've attached the video with this PPT, and I also referred some papers which is called "Attention is All You Need" from Google, and AlphaFold's original published. AlphaFold's original papers, and I also did some research in YouTube and stuff. So, and I got a demo model of AlphaFold Mini, which is done by uh, someone from GitHub. So that's it. That's it for our project. I guess I made sense. I was able to convey whatever I was trying to convey properly. So thank you.